Yeah, happiness. Very good morning. Happiness. This is Dr. Srikanth from Team MDS CONQUER. Now I'll be discussing biochemistry. So, as already discussed many times, that biochemistry is one among the list which is rarely or solidly memory based. Okay, so conceptual things will be very limited. So, it's more of a memory based, and it should be the subject of choice, and that should be taken or revised in the last 3 days for the preparation. So hope you remember that we have discussed the different types, that is the three types of porphyrins, and out of which euro is most important. They have asked a question like which is the most soluble porphyrin, that is uroporphyrin. The least is proto. The next goes is the synthesis, okay, the biosynthesis of heme. So for basic biosynthesis of heme, that is leading to the formation of ALA. Okay, the primary enzyme that can be considered as the enzyme that is required is ALA synthetase. Okay, so ALA synthetase. Okay, so this is just a combination of glycine plus succinyl CoA in the presence of ALA synthetase, leading to the formation of ALA, which occurs in the case of your mitochondria. The next comes, as we are discussing ALA. ALAs are of two types, that is the type 1 and type 2. So just do make a note about the difference where type 1 is present, what is the use of type 1 and type 2. The next comes, most of you are familiar with this CPKs, CPK 1, 2, 3. These are the three isomers that are present in the CPK. CPK helps in the conversion of phosphocreatine to creatine. CPK1 is present in the case of your brain, 2 is present in the case of your heart, and 3 is present in the case of your skeletal muscle. Most common regularly asked questions. The next goes the enzymes in the case of your myocardial infarction. That is the first enzyme that you can see in the myocardial infarction is CPK, and the last that you can see is LDH. You can remember last L for LDH. And the other enzyme that you can see is AST or SGOT. So these are the enzymes, these are the three enzymes primarily which you can see. Apart from this, there are something called as troponins. These are the markers. Okay, they are cardiac troponins. They can be troponin I and troponin T. Okay, so these are comparatively, I mean, like you can see troponins first, followed by the CPK, followed by SGOT, followed by LDH. So this is one more variation. Regularly, most of your books just stick on to the first marker is the troponins, or the next important aspect that they focus is CPKs. Okay, regularly. So apart from this, there is one more thing called as FABP. What is that FABP called as? FABP is nothing but heart type or cardiac type fatty acid binding protein. Okay, this is the one which is seen first, which is missing in most of the books. This is just an advancement of the question. Which of the following marker is most commonly seen in the case of myocardial infarction? If you don't have the answer troponins, it's better to, if you have answer troponins, yeah, you can go for it. But the advanced option is going to be FABP, that is fatty acid binding protein. You can see this again, this is a graph that is regularly asked. Okay, so which are seen first? Okay, FABPs, followed by troponins, followed by CPKs, followed by SGOT, and last but not the least is LDH. So I was talking about LDH. So basing upon this electrophoresis, your isoenzymes are going to be divided. Like basing upon the electrophoresis, there is type 1, type 2, type 3, type 4, and type 5. Type 5 is the slowest. Type 1 is the fastest. Type 1 is most positive. Okay, and type 1 is present in the case of your heart muscle. Type 5 is present in the case of your skeletal muscle. In a normal individual, you have more type 2 when compared to that of the type 1. Whenever the patient is having cardiac problems, there will be drift to the type 1. This is called as flipping. You have something called as flipping isotope. A flipping isotope of heart is regularly you'll have 2 more. Whenever the 2 is drifted and 1 is more, then it's called as flipping. And in the case of liver diseases, you will have LDH5 as the predominant one. And these branchings are important, okay, which are completely memory based. I hope like most of this stuff is covered in your PP books. Just go with this, okay. So like what is the branching and everything, okay. So do make a note about what is the branching? This is the branching. These are the branchings. Okay, so please do make a note. This is completely memory based. Make a note of these branchings. Carbohydrates are divided into simple carbohydrates, complex carbohydrates. So regularly ask which is disaccharide. Okay, so these are the disaccharides. These are the monosaccharides. The next goes, what is the use of DNA helicase enzyme? So what is the use of DNA helicase enzyme? This is an enzyme that unwinds the double helix by breaking the hydrogen bonds between the complementary. So these are the two complementary things, and these complementary things are attached by the hydrogen bonds, and DNA helicase is an enzyme which is going to break this hydrogen bonds. There is something called as endonuclease. Endonuclease is a restricted enzyme which recognizes and cleavages the particular sequence, okay, in the presence of Mg+2. Factors that are influencing the enzymes. First, the important factor is the temperature. Okay, so what is going to be temperature? As temperature increases, the velocity of the enzyme increases up to an optimum level, that is something around like 40 degrees, and then decreases. This is a bell-shaped curve. The next goes is the pH. As the pH increases, it increases and it drops. This is also a bell-shaped. This is also a bell-shaped. Then goes with the classification of enzymes. I hope like most of you are familiar with this classification. Make a note about the order. Okay, the sequence is very important. O, T, H, L, I, L. Okay, so the sequence is very important. They're gonna give you this example and they can ask you what type of reaction, or they can give you the examples. You can see NAD oxidase. Okay, you can see cytochrome oxidase. You can see D-amino oxidase. All this comes under oxidoreductases. The next one, characteristics of the enzymes. Enzyme is a protein. Enzyme is heat level, water level, and it consisting of 16% of nitrogen. So there is something called as Q2. Q2 is nothing but temperature coefficient of the enzyme. So what does Q2 means? It is a factor by which the rate of enzyme increases for every 10 degrees increasing the temperature. So as we have discussed, like as temperature increases, the rate of reaction of enzyme increases, right? So how much time it is increased? By increasing 10 degrees, how much time it is increased is nothing but Q10. And for most of the enzymes, the Q10 value is 2. The next comes is, what is Vmax? Vmax is the maximum velocity. And what is Km? Km is the substrate level concentration. Km is the substrate level concentration at which the velocity is going to be half of the maximum velocity. So Km concentration is the substrate concentration to produce the maximum velocity, half of the maximum velocity. So do make a note of these concepts. Low Km value indicates a strong affinity between the enzyme and the substrate. So to have a strong affinity, you should have low Km value. Means low substrate concentration is going to produce the maximum velocity. Km values are something like a thumbprints of an enzyme. Km value does not depends upon the concentration of enzyme. So these are the points that you have to make a note. Okay, normal reaction is going to be, you are going to have an active site, substrate is going to come and bond. This is substrate and enzyme complex. So once the reaction is done, the substrate is going to go off and enzyme is going to be normal. So there is something called as competitive inhibitor, non-competitive inhibitor. Competitive inhibitor means your substrate and your inhibitor, they have both structurally they're same, and they compete for the active site. So what is competitive inhibitor? There is some competition for the active site that is called as competitive inhibitor. The second one is the non-competitive inhibitor. The substrate and the inhibitor are structurally different and they don't fight for the active sites. As they are not competing for the active sites, it is called as non-competitive inhibitor. So you have to make a note about the differences of competitive and non-competitive inhibitor, which is a most commonly asked question. So please do be perfect with each and every line. The concepts regarding these lines were discussed when we are going with the normal classes. For now, you have to be thorough with this table. If you don't have concepts, even then mug up this. This is mandatory. If they are planning to give a question on this area, this difference is a very, very favorite area for most of the paper setters. Next, coming to the structure of enzyme. Okay, holoenzyme is nothing but the functional group of enzyme. What does it contain? It consisting of two parts. One is protein part, which is called as apoenzyme, and the second one is non-protein part, which is called as a coenzyme. And the prosthetic group is the group which bonds this protein part to non-protein part. So this is called a central dogma of the life, that is the DNA, which is going to undergo transcription leading to the formation of RNA, which is going to undergo translation leading to the form of the protein. So reverse transcriptase means DNA formed from the RNA is called as reverse transcription. So I hope most of you are familiar with this. What is the difference between the nucleoside and nucleotide, and what are the what are the amino acids that are present in your RNA, DNA? What is the difference between, here you have uracil, here you have thiamine. So next comes the double helical structure given by Watson and Crick. Make a note, the Nobel Prize was given in the year 1962. It's one of the major milestone in the case of your modern biology, and the futures of Watson and Crick are basically on bDNA. Different types of DNA are there, so their structure is basically bDNA. So do make a note, they run parallelly in anti-parallel direction, one from 5 to 3 direction and 3 from 5 to direction. Okay, so do make a note about 3.4, 0.3 nanometers. What is this? All these very important. So width, what is the width? What is the what each turn? What is the pitch of each turn of the helix? How many base pairs are present in each turn? What are the bondings? What are the sidewise bondings? What is the bonding between G and C? Number of hydrogen bonds here between A and T. Number of hydrogen bonds between the G and C. All these are very important. This particular rule is again the most common areas. Most of these concepts are very familiar to you. So we have discussed many of these concepts. We have done many problem solving on these areas. I hope you're. And coming to, I'm just going to give a brief idea. So this is a main chain. So they can give you the description of the main chain, and they can ask you the complementary chain. Okay, so what's complementary chain is, A is going to be replaced by T, T is going to be replaced by A, G is going to be replaced by C, and C is going to be replaced by G. So this is what the complementary chain. So in the question they can give you the main chain, they can ask you the complementary chain, followed by, so main chain and complementary chain we are done with. So next is transcription. So what happens in the transcription? Your RNA is going to be formed. So what is going to be RNA? So and one more thing, your main chain is in the direction of 5:3, and your complementary chain will be in the anti-parallel. It will be in the direction of 3:5. Whereas your once the transcription is done, your T's are going to be replaced by U's. That is the only thing, because that's the difference between the DNA and RNA. So A, T is replaced by UU, C is C, G is G, A is A, and T is replaced by U, and G is replaced by G. So this is called as transcription, and the mRNA sequence is ready. So once the mRNA sequence is ready, so this is a main chain, we are done with the complementary chain. Now your mRNA sequence is ready, and 3 gives rise to formation of 1 amino acid, right? So this is an amino acid. So they can ask you number of hydrogen bonds. So A and T has 2 hydrogen bonds, C and G has 3 hydrogen bonds. By counting the number of hydrogen bonds, okay, they'll give you this is X%. X is the amount of energy required to break 1 hydrogen bond. So how much of energy is required to break this double stranded DNA? So by counting the number of hydrogen bonds, you'll get the energy levels. And glucose 6-phosphate is an important compound that joins many metabolic pathways. So what are the metabolic pathways which are linked to the glucose 6-phosphate as these? And of course, you have many questions on these enzymes recently in the last NEET exam, that is AST and ALT, that is aspartate ammonium transferase. Okay, so when you compare these ratios, you can identify most of the liver diseases. If it is less than 1, then it's going to be hepatocellular damage. If it is more than 1, it is cirrhosis of the liver. If it is more than 3, it is alcoholic liver disease. And coming to, you have a question on this area also too, in the direct taken from your offline class, that is the addition of fluoride. Okay, so what happened to the addition of the fluoride? So when the fluoride is added, fluoride is going to react to the magnesium to form magnesium fluoride, which results in the decreasing of the magnesium available, and that is the major ion which is required for the activation of analyze enzyme. Okay, so fluoride is going to inhibit going to inhibit the analyze enzyme, which is basically helps in the process of glycolysis. So what happens, what is the anticoagulant that is added to this in the blood blanks is that is potassium oxalate. And what is the glycolytic inhibitor? You should prevent the process of glycolysis. So glycolysis can be prevented by adding NAF, because the F is going to react with the magnesium there, and it's going to inhibit the analyze enzyme. And do make a note, the cancer cells, they divide rapidly and they lack an excessive capillary network for oxygen supply, and the glycolysis is the main pathway by which they get the energy supply. So glutes are important, not asked in the last 2 or 3 needs, but it's a age of like in 2001 to 2015 you have many questions. Every exam has questions on glutes, and this is an important table that you have to make a note. The next comes are the phosphorylation state reactions, energy phosphorylation reaction. So this is a reaction in tricarboxylic acid cycle or Krebs cycle, and these are the two reactions in glycolysis. So this is a new modified generation of ATP. Most of you in the recent 2 or 3 days were asking so many questions related to the calculations. In most of the MCQ books, they still did not change the updated version, but this is the latest thing that is available from your Satyanarayana. So please do make a note of these classifications. Make a note about the old classifications also. If you find new classification option, go for a new classification option. If you don't find new, then you can go for an old, because repeats are repeats always. Okay, so if they're giving a repeat over this area, you have to go for old. Okay, you have to go for old, because if you don't find option which is related to the new, then you can go for the old options. What is respiratory coefficient? So respiratory coefficient is the volume of carbon dioxide produced by the volume of oxygen consumed for unit time. So it's going to be 6/6, and it's going to be 1 for the carbohydrates. What is the difference between the saturated and unsaturated fatty acids? And most of you are well aware of this, so please do make a note. And this is a classification that is very, very important. So they can ask you which of the following is a saturated, or all of the following are saturated fatty acids except. So they can ask even the questions related to the the formulas and everything are being asked in various examinations. Okay, but do make a note at least the classifications. Okay, all these classifications, what are saturated, what are unsaturated, what is marked, and everything are important. The next comes is the anion gap. Anion gap can be defined as the difference between the total number of measured cations on one side to the measured anions on other side, that is giving the anion gap. Anion gap is nothing but unmeasured anions in the plasma. So what's going to be? It's going to be 36 plus 4. It's going to be 100 plus 25. So total the anion gap ranges from 8 to 18 milliequivalents per liter. So basing upon this anion gap, most of your acid-base reactions or the acid-base relationship of the disorders will be there. Please do make a note about this acid-base stuff. Okay, so 2020 NEET doesn't have a question on this, but it's a very familiar thing that they regularly ask. I want you to go to your acid-base reactions. Okay, I mean, like we have 1 or 2 lectures on this, one taken by me and one taken by Pradhan sir. So I want you guys to go just have a look, all the normal values, your partial pressure of oxygen, carbon dioxide, bicarbonate values. There are many other values that you have to make a note, and you have to conclude what is this. Okay, so whether it's respiratory alkalosis, respiratory acidosis, or all the 4 options, how it is compensated, everything. So please do make a note. That's very, very important. So this is next comes is the catecholamine biosynthesis. So this flowchart is very, very important in enzymes. It starts with phenylalanine. In the presence of hydrolyzed enzyme, it's going to convert into L-thyroxine, and it's in the presence of again a hydrolyzed enzyme, it's going to convert into L-dopa. In the presence of decarbylase, it's going to convert into dopamine. In the presence of hydrolyzed, it's going to convert into non-epinephrine. In the presence of methyl transferase, it's going to convert into epinephrine. So all this sequence is important. They can ask you which enzyme converts this to that, or they can ask you the starting product or the ending product. Many questions were there on this particular area. Which reaction? For example, recently in one of the examination, they have asked what is the type of reaction for converting the L-dopa to that of the dopamine. The answer is decarboxylase. At glycemic index, most of you are familiar with this glycemic index, so please do read. This is a formula for the glycemic index, and do make a note. Glycemic index of a complex carbohydrate is lower than refined carbohydrate. So for a refined carbohydrate, you have more glycemic index, and these values are very, very important. So most refined and simplified carbohydrate is your glucose. So glucose will have more glycemic index when compared to that of any of these. Okay, so they can ask you a high glycemic index, low glycemic index, anything can they ask. So I hope most of you are familiar with this maple syrup urine. What are the amino acids that are present in this? What are branched chain stuff that are present in this? Okay, so please do. We learned so much related to this. Many questions in your mock examinations. So please do learn. And I hope most of you are familiar with this. Western blotting technique is to identify the proteins, and it's a test of choice for your screening test for your AIDS. Okay, and southern blotting technique is something like it's for DNA. You can remember it as South Indian DNA and North Indian RNA. Okay, so please do make a note of all these contents. I mean, most of these contents are well organized and arranged in your PP books, which are solidly memory based. So urea cycle, most of your questions will move around this area. Okay, so please do make a note the defect and the enzyme involved in it, and please do make a note about the lipid storage diseases too, glycogen storage diseases. They can ask you the type, because this is a question that is given in 2018 and 2019 NEET also. Very, very important area, favorite area for questions for the paper setters. And of course, you're well familiar with what are essential amino acids. Okay, this is a code. What are the non-essential amino acids? This is a code. Do make a note. Basing upon the metabolism, ketogenic amino acids, ketogenic and glycogenic. So what are they? Please do make a note. There is something called as semi-essential amino acid. That was the question given in the recent NEET according to few of you. Okay, if you don't find the option essential, then you can go for semi-essential as essential. Okay, so please do learn what is the why they are called as semi-essential. Okay, what are the other names of essential, non-essential? Please do go through this table is going to help you out. What is 21st amino acid? What is 22nd amino acid? What are the uses of this? To make a note. Few think sulfur containing amino acids, CMC, and acidic amino acids will have all the acids in it. And please do make a note, this aspartic acid is helpful in the root AS estimation, which is given in one of the examination. What are basic amino acids, what are aromatic amino acids, and few functional groups, okay, which I have given in the mock examinations. If I'm not wrong, in INICET mock examinations, I have given decent number of MCQs on the functional groups, including in the synopsis. So please do check that. Okay, so all these amino acid classifications are very, very important, which should not be skipped. What is this dipolar ion which has both the positive as well as the negative charge? Please do go through this. What is isoelectric pH? It is defined as a pH at which the molecules exhibit both the positive and negative having a net value 0. So this isoelectric pH is pH 6. So vitamin A deficiency, what is the first sign that is seen? What is the first symptom that is seen? What are biotic spots? And you can see how they are clinically many times given and discussed on the group. Very, very important for diagram based stuff. Okay, so please do read this. High concentration of retinol increases synthesis of lysozymal hydroxylase. And you can make a note, vitamin A, excess of vitamin A, okay, can lead to diseases related to the lysosome cell organelle. Okay, two vitamins which can be seen as a hypervitaminosis is A and D. Okay, so please do make a note about the antioxidant properties. Antioxidant properties vitamins are A, C, and E. Most antioxidant properties seen in the case of E. The next comes is the vitamin D. What is vitamin D2? What is D3? Okay, so what are the sources of these vitamins? Okay, these are called as provitamins. Okay, vitamin which acts as a hormone is preferably D followed by A. Even A acts as a hormone according to your Schopenhauer reference. So they can give you, so regularly vitamin which resembles the steroid in structure and functions like a hormone is D, and vitamin which can also act as a hormone is vitamin A because of its epithelialization. Okay, so this is a sequence that you have to make a note. What happens in skin, what happens in liver, what happens in kidney. So the consequences. Okay, so whenever the plasma calcium level is low, what happens? Okay, your parathyroid hormone increases, calcitrol increases, and leading to all these consequences. And this is a very important thing about the synthesis of heme. Okay, synthesis of heme and how it undergoes conversion to bilirubin, biliverdin, and enters the hepatic circulation and excretes in the feces. Nowadays, your hepatocellular jaundice, obstructive jaundice, different types of jaundice, and all these are the questions which moves around this concept. So please do make a note about these vitamin D deficiency metaphysis. Okay, so please do make a note about this diagram based stuff. Okay, Ragazza's spine is seen in the case of, okay, so what does it actually mean? You can see in CT scan. Then after, this is a typical thing that is called a sandwich vertebra. Okay, so please do go with the sandwich vertebra, which is seen in the case of osteopectrosis. This is also seen in the case of vitamin D rickets. Yeah, I'm done for now. Signing off, Dr. Srikanth from Team MDS CONQUER, and All the best, guys. Make a note. Biochemistry is important. Your PP books and your dental pulse synopsis. Your synopsis in your PP books and dental pulse are mandatory. Please do have a glance because this is solidly memory based. I don't want you to skip or miss any question in the final exam related to the stuff which is already being done by most of the competitors. Bye. Take care.